I remember a presentation by P.Z. Myers where P.Z. Myers was talking about how much and how the vast majority of the DNA is non-coding, how 8% of our DNA is endogenous retroviruses, how some other percent is the product of a buggy enzymatic process, how some of them is copies of copies, and this is the bulk of it. So it's something like 10% or less, or 5% only. I can't remember the number off the top of my head, but that's what we have for coding DNA.
DNA, we now know that junk DNA, the non-coding regions of DNA, function much like the operating system in a computer that is directing the timing, expression, and regulation and use of the other information or the data files that code for proteins. We know these regions regulate DNA replication, transcription, mark sites for program rearrangements of genetic material, controls RNA processing, editing, and splicing, and repairs DNA. It even regulates embryological and placental development, and even aids in immunodefense. This is just a tiny fraction of what these DNA regions actually do. The fact of multiple overlapping codes make our information systems both polyfunctional and polyconstrained, which is yet another major fatal blow to evolutionary theory. We are dealing here with a highly compressed DNA code beyond the best computer code. We write books. We don't write books that can be read both forwards and backwards. To bring this statement here about junk DNA to a conclusion, this evidence of functionality will distinguish between the two models, evolution versus creation, being discussed. For the evolution model, all differences in nuclear DNA are due to mutations. And in the creation model, differences in mitochondrial DNA sequences are due to design and mutations. When comparing DNA sequences in similar genes in different families, evolution says all differences are due to mutations, while creation says the differences were designed with distinct functionality. And this prediction involving the non-coding DNA regions, evolution says there will be much non-functionality, maybe 20%, while creation predicts functionality for most of the genome. And results from ENCODE project have suggested that at least 80% of the human genome is functional. These results are preliminary, but the trajectory is clear. Most non-coding DNA in the human genome is functional and serves in a regulatory capacity, especially since portions of the genome involved specifically in embryological development were not evaluated. However, systematic knockout experiments can only fully demonstrate this. Junk DNA is dead. Richard Dawkins in his book in 2009 said, it is a remarkable fact that the greater part, 95% in the case of humans, of the genome might as well not be there for all the difference it makes. After Richard Dawkins realized junk DNA is dead based on ENCODE, in a debate, Dawkins stated, I know there are some creationists who have jumped on it, junk DNA that is, because they think it is awkward for Darwinism. Quite the contrary, of course. It is exactly what a Darwinist would hope for, to find usefulness in a living world. So you can see him changing his mind there in light of the new evidence. The junk DNA and how long?
Minute and a half left.
Well, are going the way of the vestigial organs. They cannot be called upon to support common descent.
All right, Stephen, another objection comes from Michael Shermer in Why Darwin Matters. And he says this, junk DNA basically disproves intelligent design. He says, rather than being intelligently designed, the human genome looks more and more like a mosaic of mutations, fragment copies, borrowed sequences, and discarded strings of DNA that were jerry-built over millions of years of evolution. What do you say to that?
Well, yeah, that's a common argument we hear. If the information, if the genome is intelligently designed, why is there so much junk DNA, DNA that doesn't code for proteins that apparently has no function? And in fact, this is a strange objection because we now know that the junk DNA isn't junk at all. In fact, what we know about the non-coding regions of the genome is that overall they function much like an operating system in a computer that is directing the timing and expression and regulation and use of the other information, the data files that code for proteins. And I've actually brought with me a short list of some of the functions of DNA that have been discovered, and I thought it might be interesting for your audience just to read them. So-called junk DNA, not junk, it's functional. It regulates DNA. Here's some of the short list of functions. It regulates DNA replication. It regulates the process of transcription. It marks sites for programmed rearrangements of genetic material. It influences the proper folding and maintenance of chromosomes, and it controls the interactions of chromosomes within what's called the nuclear membrane and the nuclear matrix. So that's just 5, but I got 5 more for you. Top 10 is like David Letterman. Okay, top 10 functions of junk DNA. Also controls RNA processing, editing, and splicing. Junk DNA, which is not junk again, modulates the translation process. That's what we were seeing in that beautiful animation earlier in the program. It regulates embryological development, it repairs DNA, and it also aids in the immunodefense of the cells. So this is just a short list of 10 of the functions that have been discovered for the so-called junk DNA. It's not junk, and the whole argument against intelligent design based upon the alleged non-functionality of the junk DNA is just wrong factually.
There is garbage DNA that just doesn't do anything. There's junk DNA that doesn't produce proteins.
Actually, the fact that ENCODE has revealed that, over 80% of the genome is actively transcribed into RNA does suggest function. And the thing is, there is accumulating data suggesting further function. And that's why our direct prediction is based on the DNA language itself, because at the end of the day, we speak less than 1%. As far as I'm concerned, given the lack of gene knockout tests of the DNA language. And all you got to do is look at the history of genetics. Taylor, back in the 40s, it was 1 gene, 1 protein, 1 function. So many things have been overturned. I mean, genetically speaking, evolution is it's dead. So many things have been overturned. I mean, even human chromosome number 2, they say, arose through a fusion of 2 ape-like chromosomes. That purported fusion site is actually a functional DNA element in a human gene. So junk DNA is dead. They always say, humans and chimpanzees share genetic mistakes, but these pseudogenes appear to be functional DNA elements, not actually mistakes. As I said, back in the day in the 40s, it was known as 1 gene, 1 protein, 1 function. So many things overturned. RNA, for example, used to be underappreciated. This is decades ago biology. There's massive literature showing unexpected functions of RNA. Evolutionists, as I've said, they hope, they dream, they imagine that pond scum to people evolution is true, but it's just not true. It's pseudoscience. And our DNA code, it's a highly compressed DNA code beyond the best computer code. We write books, but we don't write books that can be read forwards and backwards. DNA can. Look at protein moonlighting.
20 seconds.
Proteins are doing things you wouldn't even anticipate. This redundancy, for example, the cell uses different redundant elements to slow and speed the process. It's a highly compressed DNA code, and that also suggests that the DNA code is also polyfunctional and polyconstrained, and any change is going to be deleterious. Well, do you know how much on a mammal, how many experiments, because the gold standard of gene functional testing is gene knockout test, right? Knock out that gene, see what the outcome is. Do you know how much percentage has been done on a mammal?
I'm not sure.
Well, very little. So it would be premature to make the claim that the majority of non-code. I understand that you agree that it's certainly active to some extent, but we would
right. Non-coding does not mean non-functional. They're not saying that. They're saying we know that DNA does this certain kind of coding function. We know of this function, and we can show that the majority of the genome doesn't do this function that we know about. That's all they're saying. They're not saying it does nothing. They're just saying it doesn't do this one function we know about. We can test it for this one function.
But the thing is, a lot of these non-coding DNA regions, say pseudogenes, for example, genetic markers, they'll use those and say that those are, genomic fossils, evolutionary leftovers to build their nested hierarchies, to build their phylogenetic trees. But they didn't predict that those genes are now necessary and required to sustain healthy life processes in the cell. Because why would a genomic fossil, like a pseudogene, for example, be active to that extent? Because that's a pretty significant role. And even in the cases where there are real broken genes, that they're actually discovered, they're often associated with heritable diseases. So are you saying that they predicted that genetic markers like the beta globin pseudogene, the supposed broken remnant of an ancient chicken gene, are you saying that they predicted that those would have important functions?
They again, the only claim they're saying is that it's non-coding. It doesn't do this one thing of coding these proteins in the way that we know it. That's all they're saying. They have no say over what it does in any other thing. So it can do lots of other things. It could do infinitely many other things. We have no idea. The only claim of the non-coding statement is that it doesn't do this one thing that we know that we can measure. That's it. That's all it says.
Well, from what I know of ENCODE, most cell types, they weren't actually tested. They only tested less than 500 cell types, and there's thousands of cell types, right?
So I'm not talking about
functions are expected from these thousands of other cell types, and that's what we're predicting based on created heterozygosity. Because if he encoded these pre-existing functional DNA differences, we would expect there to be genome-wide functionality, and not just activity, not the fact that it's just idle, but kind of like what we're seeing in these specific pseudogenes, beta globin pseudogene, for example, that they're actually necessary to sustain healthy life processes in the cell. Just like ERVs, endogenous retroviruses, very, very important in placental development, the embryo. So everything seems to be at least consistent with that hypothesis.
The ENCODE project would say that anything that has any kind of activity, whether it be conformational changes, whether it be protein encoding, whether it be MRA, mRNA, whether it be any kind of spurious transcription, anything that any detectable activity, they call that functional for their purposes. But that's not typically what it is. The functional DNA is a coding for protein DNA, and Standing for Truth doesn't seem to understand the difference or want to understand the difference.
In my new book, Zombie Science, I touch on the issue of junk DNA, so-called junk DNA, about which I wrote another book several years ago, The Myth of Junk DNA. When the function of DNA was first being worked out, biologists realized that only about 2% of our DNA, of human DNA, codes for proteins. And even then, as I point out elsewhere in the book, it doesn't completely code for protein even then. But 98% of our DNA, apparently to them, had no function at all. And so Richard Dawkins and others argued that it was just a leftover from evolution, like a harmless parasite that just hitched a ride on the DNA that actually did something. And so for many years they argued that 98% of our DNA was junk, and that this provided evidence for evolution. Jerry Coyne has argued this way. Francis Collins argued this way in a book called The Language of God. And yet, in the last 15 years, we've discovered that this so-called junk DNA actually serves some very important functions. The year after I published The Myth of Junk DNA in 2011, a project called ENCODE, or Encyclopedia of DNA Elements, came out with a whole bunch of papers documenting functions for the 98% of our DNA that does not code for protein. And the functions have just increased since then. I mean, hardly a month goes by when some other element of so-called junk DNA is not reported to be functional. Now, interestingly enough, defenders of Darwinian evolution still insist that it's junk. They say that the measures of function used by the ENCODE team, namely that it produces RNA, or it mainly, that's the main function actually, it has a biochemical function, are not really functions when you look at things from an evolutionary perspective. And since nothing in biology makes sense except in the eyes of evolution, in the light of evolution, according to them, it's still junk. So there's a controversy right now between people who study the actual functions of DNA and those who insist that evolutionary theory is true no matter what. The latter, to me, is a form of zombie science. Well, the ENCODE scientists are just good scientists doing their work to find out how something works. I don't know most of them, frankly, personally, but I don't know any of them that would be called creationists or even intelligent design proponents. So they're just doing their work and reporting their evidence. What they're not doing is they're not allowing evolutionary dogma to dictate their results. And that, to me, makes them good scientists.
Where is it getting that the vast majority of DNA is functional? Because
he runs the argument from the ENCODE project.
Well, I don't think we know. There seems to be a lot of DNA, which is deoxyribonucleic acid. That's actually the code in every cell of your body. You have about 100 trillion cells in your body. And each cell has 46 chromosomes, except for the gametes, they've got 23. Each of those chromosomes is the DNA, is the deoxyribonucleic acid. It is unbelievably complex. There's more code in each cell of your body than there is in any computer ever built by man. Unbelievable amount of information and code in there, more complex than the space shuttle. Each cell of your body is more complex. But what they oftentimes thought was junk DNA turns out to be a backup system or redundant. So in case some DNA is damaged, X-rays from the sun come down here and they go right through your body. On the way through, they just like getting shot with a machine gun. They blow your body full of trillions of little tiny holes. Sometimes they damage the DNA code because they go right through the nucleus of the cell. But the body has a backup system where some of this DNA is in there twice. It's in there redundant. You have the same thing in your car. You probably have a brake and you have an emergency brake just in case, something goes wrong. So it's just wise planning to have a backup system. I don't think we know that there's any junk DNA anyplace. It's probably just a matter of they don't know the use of it yet. What they'll do is they'll take a cell and they'll clip out a piece of DNA and say, see, it lives okay. Well, that doesn't mean it was junk DNA. I think it'd probably be impossible to prove actually there's any junk DNA. It's just a matter of our lack of understanding of what it does, or it's a backup system.
I would agree 100% with you on that one, Dr. Hovind.
How 8% of our DNA is endogenous retroviruses.
At the end of the day, if ERVs are found to have function, then it is highly unlikely that they were inserted by retroviruses. And functional ERVs would depend heavily on their position and their sequence. If they were positional and positioned slightly wrong, or they had the wrong sequence, they wouldn't be functional. The evidence is certainly on our side when it comes to this highly compressed, genome of ours. But that means no ERV, none of these endogenous retroviruses should have function.
I want to respond on the retroviral sequences topic. He's absolutely right. There are hundreds of thousands of copies of these virus-like sequences in our chromosomes of mammals and fishes and even the yeast cell. Now, what's interesting is that he's wrong on a number of points. Here's the first point. One of the first class of sequences to be expressed shortly after the first or two-cell embryo, that is from the zygote, including our zygotes, mouse zygote, to the two-cell stage embryo, first class of sequences are the endogenous retroviruses. And it was determined in 2004 is that if you knock those sequences out, you block their expression, you stop embryogenesis at the 4-cell stage. Actually, it's cited in a paper that Jim Shapiro and I, one of the papers published in 2005, where we dealt with this topic, this whole issue of why this so-called junk DNA is functional. Here's another example. What holds the placenta together is a class of proteins, and they form tethers. And the proteins that are coded are specified by retroviral-like sequences. It's different in the mouse than the ones in the human. If you look now, there are these switches that flip gene sequences on and off. They're called promoters. You can think of them as molecular toggle switches. A recent analysis of the number of these genetic switches in our genome contributed by these endogenous retroviruses, so many of them they just have to be garbage, 181,000 roughly. So, and you look at the literature, what they're doing now, and this is, the ENCODE project was published in 2007, a series of papers where they looked at not just the sequences, but the expression of sequences, how they're copied in RNA. And what they found, to their surprise, is that most of the expression is taking place in this so-called non-coding DNA. And you can find papers in Nature and Science, Genome Research, Genome Biology, Thousands of them are out there now. Go to PubMed.org again, just type in junk DNA and see what you pull up. Don't take my word for it. But what you'll find is that when they investigate these classes of sequences, as numerous as they are, they're finding a whole series of functions. Pseudogenes, for example, so-called pseudogenes, their expression regulates, yes, they don't code for proteins, that's true. Do they look like defunct copies of other genes? Yeah, they do. But you have two papers back-to-back in Nature, and what they demonstrated is that, well, those so-called non-functional RNAs, the transcripts, are regulating other genes.
A bacteria can never produce a non-bacteria. It never happens. We didn't evolve from bacteria. We're not bacteria.
You hypocrite, you teach frogs came from an amoeba.
Never, ever, ever, period, not at all, does it ever turn into something else that its ancestors wasn't.
Do you believe the T-Rex is related to a carrot? You do believe a T-Rex is related to a carrot. You believe a whale is related to a pine tree. That's a belief system, Mr. Nelson. That's not science. Don't call that a lie. I'm not telling a lie when I say evolution's a religion. It is a religion.
Has ever seen a frog produce a non-frog or a cow produce a non-cow? You can believe it if you want, and you obviously do.
Why are you calling that lie number 4? That's the truth. Nobody's ever seen a cow produce a non-cow. Now, I use the word cow. Cows produce cows, and somebody in my comment section said, oh, Hovind doesn't know anything. It's a cow and a bull that produce babies. Cow is the generic word that I think all people use to describe the cattle kind. Okay, I'm sorry. We'll call them cattle if that helps, all right? I know about a cow and a bull, okay? Anyway.
No, I obviously don't believe that.
Oh, notice the drama. No, I don't believe that. Yes, you do. You do believe the cows and bananas are related. Yes, you do. Be honest.
That is a straw man fallacy because evolution doesn't teach that and never did.
Yes, it does. I'll show you in a second.
You can't produce a secular textbook to support what you said because...
I think I can. Here's the one you're going to mention in a few minutes, the Campbell Biology. This is the 2017 edition, Mr. Nelson. And we got all the pages marked. Yes, the updated latest one. Yes, they do teach all life forms have a common ancestor, so the cow and the banana are related. That is still what they're teaching. Yes, it is.
Because evolution doesn't even permit that, and it would violate evolutionary laws.
What? Evolution doesn't permit a frog producing a non-frog or a cow producing a non-cow. Watch what they do here now. This is slick. They say, he's going to talk about going forward, cows will always produce cows, dogs will always produce dogs. Mr. Nelson, go look at your charts. Cows and dogs came from an amoeba. Was an amoeba a cow or a dog? Or did it somewhere along the line produce something other than its kind? Did it go out of its clade?
Descent with inherent modification adheres to monophyly, meaning that every new genus or species or whatever is just a modified version of whatever its ancestors were and cannot grow out of their ancestry.
Bingo! You're finally getting it. God made them after their kind, and they always bring forth after their kind. They cannot grow out of their ancestry. Whoa, there's hope, folks. There's a light at the end of the tunnel from this dimwit who thinks that cows and bananas are related. They cannot grow out of their kind. Did the bacteria that turned into a whale and a pine tree, did that grow out of its kind, Mr. Nelson? How can you be such a hypocrite and say they can't grow out of their kind when you do believe they did grow out of their kind? A few more seconds here. We got to go. I got another debate to do here. Okay.
So evolution allows new and different varieties of frogs, but it doesn't allow any frog to produce a non-frog.
You hypocrite. You teach frogs came from an amoeba. Doesn't he? Yeah. Think about what you're saying, son.
Nor for an ape to produce a non-ape, which is why we are still apes.
Speak for yourself.
Yeah, exactly. Yeah. Okay, so it was more like a bush than a tree. Okay, so it's very hard to determine the ancestry at a certain point.
Yes, it is, but you expect creationists to identify all these created kinds in your challenge. Like that makes any sense when you guys can't even do it yourselves.
But you're not saying it belongs to a different tree of life from me, are you?
Well, I think the tree of life is an artifact of some early scientific studies that aren't really holding up. So the tree, there may be a bush of life.
Right. Yes.
Bush.
I don't like that word.
But that's only in.
All right, I concede that point. Yeah.
So there is not a tree of life, and in fact, from our deep sequencing of organisms in the ocean, out of now we have about 60 million different unique gene sets. We found 12 that look like a very, very deep branching, perhaps fourth domain of life.
Right there. Let's start right there.
But disease increase is evident, just like the Bible describes. And we can show with empirical
wait, what?
evidence that there is both a fast
Wait, what did he say?
clock and a
Hey, back up about 45.
If Dr. Meyer keeps on adding his intelligent design argument there, he's retreated out of science once again. It's supernatural. It's not a testable hypothesis because he will not allow it to be falsified.
It is testable, and it's testable in the same way all historical scientific theories are testable. You ought to know about the method of historical scientific reasoning. I show in the book, Signature in the Cell, that the case for intelligent design is based on the same method of scientific reasoning that was used by Darwin and by Lyell. That method is a method called the method of multiple competing hypotheses, standard in geology, where I was also trained. And it is and the hypothesis of intelligent design is testable by comparing its explanatory power over and against its competing hypotheses, and it's also testable by comparing the predictive success of the theory